The work I'm going to present today, actually I did when I was postdoc at EMBL Heidelberg, when I worked with Martin Beck there. And the puzzle that I solved there emerged actually much earlier, something like 2 billion years ago, at the ocean floor around so-called hydrothermal vents. And the water around these hydrothermal vents was full of microbes. So for non-biologists, microbes have the genomes shown in green here, freely floating in the cytoplasm. And around 2 billion years ago, some of these microbes developed a membrane around this genome, so-called nuclear envelope, and forming a nucleus and giving the birth to eukaryotes like us, which are defined by the presence of this nucleus. And this membrane was very useful for regulating the expression of genes that are located in the genome, but also was very useful to protect the genome from foreign DNA, from foreign nucleic acids, and from viruses. But it also created a problem, which is how to transport molecules from outside the nucleus, from the cytoplasm inside the nucleus, and how to export big molecules from the nucleus. So for instance, the products of gene expression. And 2 billion years later, humans actually evolved, and they got curious how does it look like, and they developed and they actually developed electron microscope like this one. And they got curious how this problem of transport is solved, and they used such a microscope to look at the nuclear envelope. And there's a view at the nuclear envelope, and you can see what they found. They realized that the nuclear envelope is full of little channels, which they called nuclear pores, because they look like pores. And following studies found that they are actually protein complexes, so assemblies of many proteins that form a channel into this lipid membrane. And these complexes solve the problem. So they import the molecules outside from the cytoplasm to the nucleus, and they export selectively that molecules. And with slightly better magnification of electron microscopes, they could see, for instance, when they imaged the frog oocyte, then actually realized, you can see with this projector, how they roughly look. So you can see that, so each of these flowers is a nuclear pore, and you can see that they have like 8-fold symmetry with some different type of density in the middle, which is not empty, it's not a hole, it's actually filled with a special disordered proteins that make this barrier. And molecules need a special ticket, a signal sequence, to go through this barrier. But this is a very low resolution picture, as you can see, and so many scientists wondered how this looks in detail. And one of the most popular methods for looking at protein structures and molecules in detail is X-ray crystallography. And roughly how it works, so first you need to crystallize your compound or your protein. And actually, how strange it sounds, we do force proteins to make crystals to find the structure, which is strange on its own. And then they shoot these crystals with X-ray, X-ray pulses, and this X-ray diffract on these crystals, producing diffraction patterns. And from these diffraction patterns, using established methods, one can create an electron density from which we build the models. Nobody, however, crystallized the entire nuclear pore complex because it's so big. It has like 100 nanometers in diameter, 100 megadalton in size, which means that this is probably one of the biggest complexes in a human cell. What they managed, and this work from many scientists all over the world, to crystallize some proteins or domains or parts of proteins. So this is how we draw proteins. So these ribbon kind of pictures represent a peptide backbone curling and folding into some structures. And different colors indicate different proteins put together. So even though this gave us atomic representation of many of the proteins of the nuclear complex, this doesn't tell us how they are arranged in the, together to form this, this little channel. And if you probably now recognize that what actually is the next step to do. We want to put this, so these structures are our pieces, and we want to put them together. But whenever you solve, if you ever solve jigsaw puzzles, you know that if you have a picture, it's just much easier. And to get the picture, we can use other methods. So for instance, cryo-electron tomography. And this is a method that uses this electron microscope that I showed you before. And in this methodology, which is now very popular nowadays, we take a sample, a cell or purified proteins or a complex or a nuclear envelope, we freeze it, and then we shine electrons in the microscope. But in addition, we also tilt the sample in the, within the electron beam. In the end, we get many projections. So many projections from this sample, which we then combine together to obtain a sort of 3D image of our sample. And this is what, and this method, Martin Beck, so my previous supervisor when I was a postdoc in Heidelberg, decided to use to get much higher resolution structure of the nuclear pore complex. And they in the lab, they purify nuclear envelopes from HeLa cells, from human cells. They spread these nuclear envelopes at special grids, which then put into a very powerful electron microscope, which looks like the box, on the right end, with I think 5 meters high. And an example image you can obtain with this, it looks like this. So this is now 3D picture rotating in your view, and these circles indicate these pores. Maybe if you focus, you can see, so it's still low resolution, but you can see that there are several pores from different views, sometimes from the top view, sometimes at the edge on the right, you can see the side views. So we have the nuclear pores visualized from many different orientations. This low resolution, so it is pixelated not because I put a low resolution picture by mistake to the presentation. It is like this. So this is the highest resolution we can get. But what we can actually do, we can actually extract all these little circles from this image and collect many such images and align them together, classify, and from that we actually we can reconstruct something like this. So now there's a 3D image of these pores that you've seen at much higher resolution. So there's still a 3D picture, and you can see this gray density, which represents a protein mass. Everything what was flexible and moving around is not visible. So it looks like it has a hole, but it doesn't. So it's, in fact, these are these disordered proteins that are moving around, but you cannot see them because they got averaged out. And this, whatever color you can see from the distance, this brownish mass is a part of the nuclear envelope that is present in the reconstruction. And there is a well-established computational technique to take those structures, for taking such atomic structures and fitting into electron density maps. And this is, for instance, a map of another complex and, with a well-defined crystal structure fitted to the map. But for the nuclear pore complex, the resolution of our image was 23 angstrom, whatever it means, it's low. So it's kind of equivalent to having a very blurred image on the box. So we needed some more data, and fortunately we have another tool, and we can call them molecular rulers. So we have special compounds called crosslinkers, which we can add to proteins or protein samples, and they covalently link. They can covalently link different parts of proteins, or between different proteins in the same complex. We call this crosslinking mass spectrometry. Long story short, we throw a crosslinker on protein, so for instance here, and then it crosslinks, like makes these links, and then we digest this protein complex with a special enzyme, which cuts proteins into pieces. But if the pieces were linked, they actually stay linked after this digest. And then we use another tool called mass spectrometer to identify this peptide sequence of these linked fragments. And from that, we can find which exact regions were cross-linked. And the output of such a procedure looks like that. So each of these vertical bars is a protein or protein sequence, and each line indicates which parts, which residues, so-called residues of proteins got cross-linked. And for us, it provides a very useful information which regions should be closed. So for the nuclear pore complex, we had the pieces, we had this blurry image on the box, and then we had some instructions how to put these pieces in this picture to satisfy these crosslinks. This is not easy to do manually, so this is where I come to games. So my background is computational bioinformatics, and I joined the group to develop software that can take all this data and put it together automatically. And this is a picture of a computer cluster because, as you can imagine, these are pretty big combinatorial problems, and we need high performance computing to address that. I don't show you the program, the code, or the pipeline I developed, although I'm very proud of it. I just show you how more or less it works. So this is the trajectory of an optimization that I run using my software. This optimization takes the structures, so these color balls around the nuclear pore are proteins or protein structures put it randomly in space. And in the middle, you see this nuclear pore complex. And this simulation would show how so-called inner ring, so inner part of this complex was modeled using this procedure. So the procedure would now start placing these structures in the map in various random different combinations, but each time trying to improve how the structures fit the map and these crosslinks, these distance restraints in the same time. And it runs. You can see that some sub-unit proteins stabilize, some still search for the place. It takes time, but in real life, it takes even longer. It's a fast-forwarded movie. So we are still, I think, waiting, for instance, for this. Now the last sub-unit jumps in, and there is some final optimization, and the structure stabilizes. So in principle, what we actually did, we actually did an equivalent of building a robot for real physical puzzles to do it for you automatically if you ever wanted for any reason. And in the end, what we obtained is the structure of the scaffold of the nuclear pore complex. So it's definitely not an entire complex. It's like maybe 70% of the mass. So we still missed these flexible parts. We learned a lot of interesting biology from the structure. Made very significant advance in the field, actually, because it was the first time you could actually look at the structure at near atomic resolution. And for us, the most amazing was that we could understand how it is formed from these pieces of the puzzle. So here, for instance, you see some proteins, and we could understand how do they bind to this nuclear envelope, and then how, with the other proteins, it makes a bigger piece, so-called Y complex, which then repeats 8 times, and I told you before, it has this 8-fold symmetry, and again 8 times, and then does the same at the other side. And then what we then later, also what we realized, that actually this inner ring is also made kind of similarly out of these kind of modules, which also then repeat 8 times. So for the first time, we actually understood what's the principle governing the architecture of this complex, and having many implications in understanding the mechanistic of the complex and also how this complex emerged in the evolution. So I hope I convinced you that solving puzzles in molecular biology is very useful and it works. We of course call it differently. We call it integrative structural modeling. So that's what I put, I think, in the title for this presentation, because we integrate many different data sources using computational modeling methods. And this is what I do at my group, in my group at EMBL Hamburg and CSSB. So first we continue asking questions how different protein complexes are organized. So we continue working on the nuclear pore complex, but we also work on other complexes. And we also work on improving our computational methods to model faster and with better precision and with better accuracy. We also started new lines of research, so we realized that we are quite good in integrating data and also analyzing the uncertainty in the output models. So we're now trying to transpose or transfer this experience to new avenues in so-called systems biology, where we try to integrate various types of data, not only structural data, but also data from genomes or from sequencing and proteomics, to model entire pathways in cells, and also how viruses interact with these pathways, and also how viruses interact with these protein complexes that we have in our cells. And in future, we still hope that actually the puzzles that we now solve, so the protein complexes, would actually become pieces of much bigger puzzles, like for instance, entire cellular landscapes or entire cells. So here I think you cannot really see, so I explained this is a part of a cytoplasm, nuclear pores, nuclear envelope, and I think a mitochondrium in the field of view. Because these days we actually now have methods that allow us to visualize complexes within cells. So there's a revolution in in-cell cryo-electron tomography and in sample preparation techniques. We have so-called super-resolution microscopy, for which the Nobel Prize recently was awarded, which allows you to mark proteins with colors and visualize exactly where they are, sometimes at single molecule resolution. And we also have, so the crosslinking I showed you was performed in vitro with purified proteins, but now actually it also works in vivo, so you can add this crosslinker to cells and get thousands of crosslinks, so 1,000 of contact points between proteins in cells. So my hope, and also for my lab, is that we can use these integrative modeling methods to actually start putting this these now bigger pieces into even bigger picture to understand how cells or cellular landscapes or organelles work at the atomic level. And with this, I would like to finish my presentation and thank you for your attention.